All right, I'm going to be talking about a project that I started with my dissertation research at UC Davis with my co-authors, my advisor at Davis, Sharon Lawler, and my supervisor here at Redwood Sciences Lab, Hart Welsh. And okay. The project is, as far as dissertation projects go, probably one of the more collaborative dissertation projects. And so while this is happening, I would like to bring that up and mention that there's been 30 field assistants that have been out collecting this data. I've gone to a lot of experts in a lot of different fields for advice, and they've taken a huge amount of their time. People like Bill Rainey in Berkeley, and then Klamath Bird Observatory, RSL folk. And so here's the title slide. Top on the list are Fish and Game. Betsy Bolster secured funding for the critical first year of the project. That allowed us to prove that we could do it and then cascaded into more funding. So that was really key. Ed Pert secured the signatures for fish removals. So that was really important. And then the Region 1, Fish and Game Region 1 supported the project, which is the only way it could happen, and also provided gill nets and other equipment. RSL I mentioned, just an amazing amount of brain power there. The Forest Service Weaverville office, every 2 weeks we take over their front lawn. Bill Rainey I mentioned, and the field assistants, and then the funding sources on the bottom. So the study is looking at, I guess I missed one. I'm going to talk about the overview of my study, let you know what we're doing out there, and then I'm just going to focus down on Rana cascadae, given the meeting. So what we have, I'm going to talk about lakes in subalpine, but this would also apply to alpine or the majority of lakes in the western United States. We have aquatic insects and amphibians, and these groups are similar in that they both have a larval aquatic stage, and then they metamorphose into the terrestrial landscape and are prey for taxa such as birds, bats, snakes. And these groups are really key because what they're doing is transferring energy collected in the aquatic landscape and distributing it to the terrestrial landscape. And ecologists have called this transfer ecosystem subsidy. So in this case, it's from the aquatic to the terrestrial landscape. These arrows are energy flow. Oh. Okay, oh well. When we introduce trout into these lakes, we know that they have some sort of predatory effect on insects and amphibians. So they're absorbing some of the energy from these groups. And, oh, I keep going backwards. And so one question that a lot of researchers are now grappling with is, are there indirect effects of trout on even the terrestrial landscape, the predators in that terrestrial landscape, because they mute the energy flow out of the system? Okay, so what our goals are to look at the effect sizes of these arrows, sort of the width of the arrow, and then also the indirect effects of trout on not just the direct effects on amphibians and insects, but also the indirect effects on the terrestrial predators. So to do this, we started a 16-basin replicated experiment. We picked 4 fish feed control basins, and then 12 additional basins that Justin Garwood was really key in picking out. And then we blocked those basins into 4 groups from north to south, and then randomly selected which ones were going to have fish removals, which ones were going to be stocked every year with fish, and then also a category where we suspend stocking in those lakes from the beginning of the project. So our study area is Trinity Alps, which has been mentioned before. It's in Northern California, kind of triangulate between Arcata and Redding and then go up from there. It's part of the Klamath-Siskiyou range, and we focused on the east side where the lakes really are, and went all the way from the north to the south. And then these are the 4 blocks and the lakes. And so up in block 1, let's say this is a suspend, stock, remove, stock, and control. And that happens in all 4 of those groups. Here's just some examples of what our lakes look like. Okay, so to get at these goals that we have, we use a lot of techniques out in the field. So first I'll talk a little bit about the insect sampling. We use sticky traps, and we set them up in transects along different distances from shore. The goal is to get an idea of the proportion of the available food for the terrestrial predators, especially the birds and the bats, that comes from an aquatic source versus a terrestrial source. We're using emergence traps to look at the abundance, the size, the composition of the insects that are actually emerging out of the lakes. We use exuvia plots that's focused specifically on odonates, and we can collect in a set area the exuvia, all the exuvia that we can find, and we can get a relative abundance, and then we can also look at the actual size. We can measure these exuvia, and we can get the exact size of the larvae when it metamorphosed to see if there's any difference in lakes with or without fish. Also, we conducted benthic sweeps. Amphibians, visual encounter, some other people have mentioned, and mark recapture using pit tags. And it's not just Rana cascadae, we're also looking at the suite of amphibians that are in there. And the Tricha granulosa, the rough-skinned newt's not on this, but long-toed salamander, Pacific tree frog, Cascades frog, and western toad. And finally, the indirect effects group, we chose birds, bats, and snakes as our receptors that we were going to look at. We do point counts and area, time-constrained area searches for birds. We do mark recapture, pit tagging again, and then VES and stomach palpation for the snakes. And bats, we use these remote acoustic detectors, anabats, and do dusk visual surveys as well. So now that I've told you what we're doing out there, I'm not going to talk about it. And I'm just going to focus on our results that we've got so far for Rana cascadae. So some of these graphs are going to look somewhat the same. Each of these points this is our first survey in 2003, and that's our 6th survey in 2005. The error bars are standard error, and so each point is the group of control lakes, the group of suspend stock lakes. So this is our control. One thing to note is the huge variability, even in the control. And that's an issue I'm going to be grappling with. I don't know, we're all probably going to be grappling with for a long time. But this orange bar is our removed lakes. And we removed fish from these sites in 2003 in the fall. So we got 1 year of pre-removal data, and then so far 2 years of post. And then the greener colors are the suspend and stock lakes. So here's larvae. We're starting to see a little bit of an increased blip in our removed lakes. And then this data summarized down to both larval and metamorphs. We see an increase in 2004 and a pretty much similar size, population size in 2005, whereas stock and suspend are staying fairly low, and the control lakes are significantly higher. And adult counts, the control lakes again, and then here's our removed sites, and we're seeing an increase in adults, and it's really starting to increase this past year. Whereas the suspend and the stock lakes that still have fish in them are staying consistently low. And then summarized in the annual data, control again higher. In 2003, the removed stock and suspend were all equal. And in 2004 and 2005, the removal sites have gone up. And I'm not showing any statistics here. We have run some repeated measures of ANOVAs on these. We do see a treatment effect, a year effect, and a treatment by year effect for metamorphs and subadult adult frogs. We haven't seen it in a treatment by year effect, which is what we're really looking for in larvae yet, and I think it has to do with the variability. But really what we want is that recruitment coming out, so I'm excited about it. Oh, I'm backing up. Okay, some questions that we're also looking at are what's going on within these basins. Is there changes? Are there differences? One way of looking at it is what is looking at the lake compared to an associated pond that doesn't have fish. So we have in half of our sites, we have one main lake that can hold fish and either does or doesn't, or fish were removed. And then we have another pond that's adjacent to that lake that doesn't, that is perennial, but isn't deep enough to support fish. And so do they use these sites differentially, given whether there's fish in the lake or not? And the reason is sort of getting at, is there a behavioral avoidance of the lake? And we definitely do. In the control lakes that have the lake without fish in it, about over 80% of the frogs found in any one survey in that basin were found at that lake. And about less than 20% were found at the associated pond. Versus in fish containing lakes, it was less than 40% of the frogs in any given survey were found at that main lake, and 60% were found at the associated pond. Adams Lake here is one of our removal sites with an adjacent pond right next to it. And immediately we're seeing we pulled over 500 fish out of this little tiny lake. So it had a ton of fish in it. And in the first year, less than 20% of the frogs that were seen during the surveys were found at that lake, compared to the pond where 80%. And then the following year, it jumped up to close to 40, and now it's over 50%. So they seem to be quickly realizing there's no fish and staying in the lake. I'm going to switch gears a little bit and look at the pit tagging data. And I just got the clean version of this last week, so this is definitely preliminary. So I wanted to look at, this is a way of looking at recruitment. So we're out there all 3 years, and whenever we can find a frog, we're pit tagging it. So I want to look at the proportion of the frogs we're pit tagging in any year that were tagged at the control lakes versus the stock versus the suspend, etc. And of course, there's majority of frogs are at the control sites, so the majority of new tags that are being used are from these control sites as well, over 50% every year. And then in the remove sites, we had less than 10% of the new frogs being tagged were in the remove lakes, and at this point it's jumped way up. And because it's a percent of the tags we're using, something has to give, and that it's decreasing in the stock and suspend lakes, the relative proportion. And so, and the actual number in 2003, in the removal sites, we tagged 27 frogs, and that means we found 27 frogs. By 2005, last year, we tagged 123. So we are seeing a lot more frogs coming in. And that made me think, well, are these frogs, where are they coming from? And if they're recruiting from the nearby area, the population there, meaning that more successful recruitment given fish are removed, then they would be smaller, likely young adults. And in 2003, the size was exactly the same across the different studies, the different treatments. And in 2004 and 2005, it was significantly smaller in the remove lakes. So I think there is some recruitment, some increased recruitment in the removal lakes. So the preliminary conclusions that I'm going to make here are that Rana cascadae populations appear to be increasing in most of the removal lakes. It appears that recovery is driven both by immigration from probably neighboring areas and recruitment. And then you'll have to stay tuned for the rest. And that's it. And this is just some pictures from the field seasons. There's a lot of people in the room that have worked on it, so you can find yourself. Are there any questions?